Alright everybody, hello. The goal of this video is to explain to you how to do a monohybrid cross so that you can basically use this as a reference when you go to work on problems. So first off, let's just define a monohybrid cross. So a monohybrid cross is a genetics cross that's tracking one gene through a generation. So it wants to basically make a prediction about what the offspring might look like. So let's just define that. It's a cross that tracks, I guess, the inheritance of one gene. So we're just looking at one gene here, and every gene has two potential versions in Mendel's world. So in this case we're going to be looking at pea plants, and the two versions of the gene are either tall, which is represented by an uppercase T, or short, which is represented by a lowercase t. So what we have here is just a sequence of steps you can use. In the beginning you want to always go back to these steps, but as you get more comfortable you can kind of move more quickly. But for now we'll do everything out step by step. So in this case we have a genetics problem we're going to work with. So it says a tall pea plant is dominant to short, so that's why I use the uppercase T for tall, and we want to mate a homozygous tall male crossed with a homozygous short female. What will be the genotypic, so the genotype and the phenotype ratios and percentages. Now make sure before you do these you know what homozygous and heterozygous are, and you know what alleles are, and you know what a genotype and a phenotype is. You got to get those vocab down. So first things first, let's write out our parent genotypes. You always want to do that first. So the male is homozygous. Sometimes I underlined it. The male is homozygous tall, so that would be one uppercase T next to another. So those are our two versions. Remember, every genotype always has two versions because each organism has two parents. So those are the two alleles that it inherited from each parent. The female is homozygous recessive, so two lowercase t's. Now what we need to do is distribute, so we're step 2, distribute the alleles into the sex cells. So remember, when sex cells get made, it takes the amount of chromosomes and it cuts that number in half. So same thing here. One T is going to end up in this sperm cell randomly, and the other one is going to end up in this one. So they get separated. Same here for the female, except they're going to make eggs. So the female, the gametes are eggs, and the male, their sperm. So Mendel, we now know that this is actually meiosis, but Mendel didn't know that. He just called this, once he figured it out, he called it the law of segregation. And it just basically explains how it's possible for the alleles to separate. He didn't know that it was happening during meiosis. We now know that, but he described it. So the next thing we want to do is basically set up a Punnett square. The Punnett square is going to give us a visual of the possible sperm and egg combinations, basically what the genes and the physical traits of the organisms might look like. So let's just set up the square. Oh, those are uppercase. These are lowercase. And now we have to show the possible combinations. So this sperm meets this egg. We'll drag them into that box. This sperm meets this egg in this box. Always put the uppercase letter first. This one meets this one, and this one down, this one over. That. So now we have our next generation. Remember, we started with the parent generation, P generation. Now we're looking at the F1. So what did the F1 give us? What are the potential genotypes and phenotypes? So first things first, the genotypes, which remember is what we cannot see. That's the letters that we're using to represent the alleles. So big T, little t is the only possible combo. So put that, and it is 100% chance that these two parents will make big T, little t. Next, the phenotype, the physical trait. This would be tall, right? Because tall is dominant over short. So they're all tall. That's the physical trait, the phenotype that I see, 100%. And in here we can really start to see the law of dominance. So basically the tall allele masks or covers the short allele is kind of what it looks like. So did we need a Punnett square for this? Not really, right? Every time you see a parent that is homozygous dominant, homozygous recessive, you know you're going to get the same thing. You're going to have heterozygous every time. So whenever you see this times this or any, cross with that, you're always going to get big T, little t. There's no other option. So they're going to be all tall in this case. Now, let's go to the next step. The next thing we want to do is kind of follow what Mendel did. We're going to take two of these offspring and cross them. So the next piece here, it asks us, okay, if two of the offspring from the first cross, the one we just did, were mated, what would be the resulting genotype and phenotype ratios and percentages? So same thing. First thing, parent genotypes. We're crossing two from the last cross. So two heterozygous individuals for tall. They're carrying the short allele, but they're not showing it. Segregation happens, right? One allele here, same deal here. Sperm and egg, and then put them on our square, cross everything, so all the potential possibilities for the offspring, that sperm, that egg, that sperm and that egg. Oh, whoops, I made a little mistake. There we go. This one is a lowercase t, remember this is lowercase t, and this one and that one. So here's our two alleles recombining, right? Fertilization happens, and then we need to lay out all of our possible genotypes. Looks like we got all three possibilities here for the offspring. Now for our ratios. 1 out of 4 are tall, or homozygous dominant. 2 out of 4 are heterozygous, and 1 out of 4 are homozygous recessive. So we have a 1 to 2 to 1 ratio. I think of these as 4 quarters in a dollar, kind of like 25% each. So 25% chance of this in the offspring of homozygous dominant, 25% chance, or excuse me, 50% chance, right, of 2 out of 4 of the heterozygous, and 25% chance of the homozygous recessive. Now we only have 2 options for the phenotype. This is just one gene, and it comes in 2 versions, tall or short. And of the tall, wherever there's an uppercase T, we know they're tall, so 3 to 1 short, and 75% to 25%. So we have a 75% chance. So if these 2 plants made 1,000 offspring, about 75% of them would be tall, and about 25% of them would be short. So that's the prediction that Mendel was able to make, and kind of the power of what he did. So use this as a reference to complete other problems. You can stop, pause, whatever you need to do. Make sure that you come back to this if you're having any issues crossing any of the questions ahead. So hope this was helpful. Email if you got any problems, me or Miss Paulison.